Let's look into paper 51, May-June 2016. Question 1. Figure 1.1 shows some of the plants growing in a pond and on the land around the pond. So you can see this is the water plants and this is the land plants. Some students decided to investigate the changes in the distribution and abundance of species of land plants. So distributions and abundance of species of land plants is the dependent variable. It is the variable in the experiment that is going to measure at different distances from the edge of the pond. So this is the variable in the experiment that is manipulated. This should be the independent variable, distance from the edge of pond. Students started their investigations at the plants growing next to the water, as shown in figure 1.1. Plants growing next to the water refer to the land plants. So in this figure, you can see the water plants here in the ponds, and this is the start of the investigations next to the water. A1. State the independent and dependent variable in these investigations. Independent variable will be distance from the pond. Dependent variable, it is the variable that is measured or tested. This is the distributions and abundance of species of land plant. Describe a systemic sampling method the student could use to find out how the distributions and abundance of plant species change as the distance from the edge of the ponds increase. Your descriptions of the sampling method should be detailed enough for another person to use. To do systemic sampling method, you can perform a belt transect, where you can place the quadrats at regular interval level along the transect. First, use belt transect as sampling method. Second, choose a site within the study area for you to place the rod of the measuring tab in a straight line. You place the measuring tab in a straight line from one end to another for a distance of, let's say, 20 meters. Minimum distance for the measuring tab to place in straight line is 10 meters. Next step is where you can place the quadrant. Place quadrant at regular interval along the line, for example, every 2 meters. Use the same size of frame quadrat for each interval. For example, each of the quadrats should be 1 meter squared. To help to identify the types of species found in the quadrat, you can use photograph, apps, or nature guides. Use photograph, dichotomous key, apps, or nature guide to identify different species found in the quadrat. Next is need to do calculations. To to check for the abundance of plant species. So it's count percentage cover of each species at each distance along the line. After you have the percentage cover, you need to use the cover abundance scales or Brand-Blanchard scales to measure abundance. Precautions. Extra precautions need to be taken to avoid miscount of low-growing species. To increase the reliability of the experiment, repeat the transect 3 times. Repeat the transect at different locations for 3 times. Safety of the experiment. This is a low-risk experiment. Wear gloves during sampling to avoid direct contact to plant that will cause allergy. Or wear masks if allergic to pollen. Wear covered shoes and protective clothing to avoid injury.